for allowing me the opportunity to present the work from John Mannick and Lederer's lab in Boston, Massachusetts. In 2001, injuries quickly cause of death in the United States. Approximately 20% of deaths after injury are from infection and multiple system organ failure. In these patients, the immune system plays a critical role. Injury reduces the imbalance of innate and adaptive immune system regulation. Initially following injury, there's an increase in the innate immune response, causing the production of pro-inflammatory cytokines. If this is excessive, it can lead to multiple organ failure. It can also lead to a phenomenon called the two-hit model of injury, which I'll go into more detail later. At the same time, there's a counter-inflammatory response by the adaptive immune system, which if excessive may lead to immunosuppression. Treatment of the patient at this stage is largely supportive. As well, immune homeostasis returns and the patient will recover. The two-hit model of injury was described by Morgan in 1993, where it was observed after numerous clinical and scientific studies that traumatic injury confined the host, such that a later, otherwise innocuous secondary inflammatory insult precipitates and exaggerating the systemic inflammatory response. Previous work from the lab and work from other investigators have described that injury may or augments the innate immune inflammatory cell profile. GR1 macrophages have been described as myeloexpressive cells, immature myeloid cells, or inflammatory macrophages. They've been shown to accumulate in mouse tumor models in bone marrow, blood, and spleen. They're defined by marking the CD11B with GR1 and differentiated to F480 cells. I'll show here that we have right beam sustained GR1 macrophages from burn injured mice, which are characterized by clearing around the cytoplasm. We hypothesized that GR1 macrophages are involved in the development of the pro-inflammatory stage and the two-hit response following injury. We had a number of experimental questions. Does injury have an effect on the GR1 macrophage population? Are inflammatory cytokines expressed by GR1 macrophages? Does injury change the macrophage response to Taylor force stimulation? Do CD4 T cells influence GR1 macrophages? And lastly, does blockade of these macrophages change the TNF expression following MRCS challenge? We did this by doing a two-hit model of combining burn injury with LPS, and in this set of studies we used in vitro LPS. Black 6 mice were randomized to sham burn injury, and day 7 following injury, killed and spleens were harvested. We carried out surface stains for GR1 and macrophage markers for expansion of cells, and we also carried out in vitro stimulations with LPS, followed by addition of Feldin A, which is a Golgi blocker, to get a snapshot of the TNF expression in the cell at that point. In brief, the burn injury model involves anesthetizing mouse with ketamine and xylazine, placing them in an insulated plastic mold, and giving them a 90-degree second burn for 9 seconds, which results in a 25% of full thickness anesthetic burn. The model is associated with 5% mortality and it simulates the immune assault seen in humans following major injury. All work from the lab was approved by the Harvard Standing Animal Committee. Our results show that burn injury causes significant expansion of splenic GR1 macrophages. On the y-axis, we have percentage of GR1 macrophages, and on the x-axis, we have our sham mice and our burn mice at day 7. And we show that compared to the shams, there's a significant expansion in the burns. The same bit is represented by the low cytometry dot plots, where here we have the sham injured mice, and here we have the burn population. We show that compared to the shams, this is a significant expansion in the burns. We also showed that in vitro stimulation of LPS results in increased TNF expression in the splenic GR1 macrophages. On the y-axis, we have our percentage of TNF expression, and on the x-axis, we have our GR1 positive macrophages and our GR1 negative macrophages, which represent good macrophages. Again, compared with shams, burns express more TNF. However, overall, compared with GR1 negatives, the GR1 positives express more TNF. Previous work from the Ashton lab showed that injured mice which are lacking CD4 cells or rag knockout mice develop a more pro-inflammatory response following injury. Our next set of studies was to address whether CD4 T cells influence GR1 macrophage activity. To do this, we randomized wild-type mice or CD4 knockout mice to sham burn injury, and at day 7, mice with those spleens were harvested. Again, we carried out surface stains to measure expansion, intracellular stains following in vitro stimulation with LPS, and lastly, we carried out cultures to quantify the amount of TNF produced. Our results for expansion show that, again, burn injury causes significant increase in splenic GR1 macrophages. However, the absence of CD4 T cells did not appear to influence this. On the y-axis, we have our percentage of GR1 macrophages. On the x-axis, we have a wild-type sham burn injured mice and a CD4 knockout sham burn injured mice. And we show that compared with shams, there's significant show that addition of LPS and increased TNF expression. This row is a challenge for mice in this bird. These mice were not stimulated with LPS, whereas these mice received, compared to shams, birds expression of TNF. This is the same data represented graphically, where on the y-axis is our TNF expression, on the x-axis we have the GR1 positive macrophages and GR1 negatives. Again, compared to the GR1 negatives, the GR1 positives express overall more TNF, and compared to shams, birds express more TNF. However, there's no significant difference between wild-type and CD4 knockouts. We then carried out cultures of cells to quantify the TNF production and analyze this using cytokine beta assay. We showed that compared to the shams, burn-injured mice produce more TNF. Our final set of studies was to address whether blocking of GR1 macrophages in vivo led to reduction in TNF expression following LPS. To do this, we randomized mice to sham burn injury at day 7 and we treated them with blocking antibodies to GR1, which is 1A8. As controls, we treated another group of mice with control immunoglobulin. These are dot plots which validate our studies. This is the population of cells interested in the control treated group, and this is the mice after depletion of the antibody. At day 10 following injury, we challenged the mice with intraperitoneal LPS, and at 30 minutes sacrificed the mice and harvested spleens into media containing Felton A to get a snapshot. We showed that functional depletion of GR1 macrophages causes a reduction in TNF expression from an LPS challenge. On the y-axis, we have a percent TNF. On the x-axis, we have a wild-type sham animal which should not receive any LPS which is treated with PBS. And we have our control immunoglobulin treated animals which show the same burn-induced increase in TNF production of the LPS. However, the animals that were depleted with GR1 expressed less TNF, particularly when we compare the IG burn-treated group with the 1A8 is significantly reduced TNF expression. To summarise, burn injury causes a significant increase in percentage of GR1 macrophages and primes them to produce higher levels of TNF alpha in response to TNF stimulation. The absence of CD4 T cells has no significant influence on the burn-induced increase in GR1 macrophages, and we showed that blocking of GR1 max caused reduction in TNF expression following LPS challenge. Clinical implications of this work may allow us to understand the source of thermodynamic cytokines and the possibility to identification of potential targets to modulate immune response to injury. And that is Jim Lederer and John Ryan.
I hope you read that very well, so a lot of complex data. Were the CD4 knockout mice class 2 negative? What was the nature of the knockout?
I'm not sure. I'm not sure. We had a problem with CD4 knockout mice. So we referred to NHC class 2 knockouts.
Yeah.
They weren't NHC class 2s. No. We also controlled and showed the data that we depleted whole wild-type mice, sham burn wild-type mice and depleted them with CD4 cells and got the same profile as control expression.
Right, so yes.
You mentioned it's quite a dynamic situation. Why did you choose the 7-day time point?
We looked at 4 different time points. We looked at day 1, day 3, and day 7. And we've seen in previous studies that macrophages tend to be most shown to the later expansion. And when we looked at the TNF profiles in the individual LPS, at day 1 there was a significant increase in the burn population towards their increase in TNF expression. So that's why purposes it shows in this data.
In terms of injury, how that would that relate? Because obviously it sounds quite late to me.
Well, the treatment conditions which developed in the first 10 days of the injury, that an issue isn't it? Someone comes in after a car crash, and then a week later they get an line infection, and then develop a response to that, and get some response to pneumonia. So we looked at early time points, but most of our effects, we see most effects sort of at day 7 to day 10 after injury. We see this in mortality studies as well.
Can I just challenge on this sort of priming thing? So priming refers to get a heightened response to the second time. I don't think you've got any evidence that you get in mice, LPS, for example, you get increased mortality.
We did numerous mortality studies where I also did some mortality studies where I tried to deplete cells, but the problem with antibodies for the native immune system is a huge cost over, and you get the same receptors are expressed on CD8 and memory T cells as well.
And then we're out to interrogate it. Probably your match.
Like one of my colleagues is working on the cell side of things because I worked on the innate side of things.
Okay, well, I think you've asked all the questions.